I am Yoshiki Tanaka, and I'm belonging to Makoto Miyata Group in Japan, same as Mone-san. And thank you for giving me the opportunity to introduce you about my research. Today, I'll talk about the behavior of MreB proteins causing Syn3B swimming. Spiroplasma educatus is one of the Mycoplasma species, and it swims by switching its helical cell body left-handed to right-handed and right-handed to left-handed. Previously, our group reconstituted this motility by introducing MreB 4 and 5 in the Syn3B with no motility. MreB 4 and 5 is the actin homolog protein. The mechanism how these two proteins cause motility is unclear, so my goal is to reveal it by discovering MreB dynamics in live cell by fluorescence observation and cell experiments. For this purpose, MreB 5 was labeled by M. Cherry. Before going to the experiments and result, let me introduce some advantages of using minimal cell for cell biology. It is advantageous for easy transformation and small cell size and limited number of large complexes. Limited number of large complexes make it easier to consider about the molecule dynamics inside a cell, and small cell size is suitable to track single fluorescence. Okay, I go to result. The MreB 5 fluorescence of moving Syn3B can be divided into three groups. In first one, the cell exhibits fluorescence and motility. And in second one, only one side of the cell exhibits fluorescence and motility. And in third one, full cell exhibits fluorescence, but the rod-shaped part looks stiff. From these observations, it seems like that maybe 5 existence and cell helicity are needed for movement. I go to result 2. One of the common characteristics of MreB is that it polymerizes into filamental structure by ATP binding. To find out if this property is related to movement, I investigated the effect of A22, which is a polymerization inhibitor of MreBs. Normally, MreB become able to polymerize by conformational changes due to ATP binding, but A22 inhibits this conformational change and disable the polymerization. Then, what happens to the cell when A22 has added? Please watch these videos. Leftmost one is the cell before A22 addition. Next one is the cell after A22 addition. Please note this part, this helical part. Is the helicity and motility has disappeared, and the fluorescence remained where the motility has existed. Next, I removed A22 from this solution. The helicity recovered, and the motility also recovered. And the fluorescence were seen at the moving part. Okay, like this, helical moving cell has straightened and stalled after addition of A22, and helicity and motion recovered after A22 removal. Yes, this result suggests that the ATP-dependent interaction of MreBs contribute to sustain helical morphology and to produce force. So the next question is that what exactly is this interaction? As for the motility mechanism of proteins belonging to the actin family, it is famous that they polymerize at one end and depolymerizes at the other end. For example, treadmilling. In order to investigate whether this motility mechanism involves large-scale replacement of subunits, I did observations using the fluorescence recovery after photobleaching, which is called FRAP. In FRAP, laser has spotted to the part of the fluorescent area, and fluorescent molecules in the area has bleached. If these molecules are moving freely, the bleached molecules and non-bleached molecules should be mixed, and the fluorescence of this area will gradually recovers. The molecule diffusion rate can be calculated from this fluorescence recovery. In my experiment, one side of the helical moving cell and straight and not moving cell has bleached, and the change of fluorescence intensity has plotted for 60 minutes. Actually, the error is high because of the cell is small and moving, and it has not been modified for few drier numbers because of some experimental difficulties. But, however, it can be seen that fluorescence has not recovered in both motile and non-motile cell for at least 30 minutes. This recovery speed is slow compared to the MreBs in other species. So this result suggests that MreB5 don't move much while moving and not moving. I consider the mechanism which can explain these results. Please notice that this is just one possibility. As found in A22 experiment, the cells are straightened when there is no interaction between MreBs. When the interaction is restored, MreBs can make curvature. The change in handedness due to ATP-dependent structure changes can be explained by considering that MreB5 is attached to MreB4, like this image. When handedness changes, it is presumed that there is no large-scale replacement between the aligned MreB5 and free MreB5. However, there is no evidence for the localization of MreB4 as described here, so I'm currently trying to fluorescently label MreB4. And I also aim to apply experimental methods that can trace fluorescent molecules in more detail. That's all for my research. Thank you for listening.
The talk is open for questions. Yoshiki, one thing that dazzles me with the microscopy in your group. So obviously you can't be shining the light at high intensity on the cells for the duration of these experiments. I assume that this is an on and off thing. Otherwise, it seems like you would damage the cells. But how do you, in these motile cells, how do you keep them from floating away?
Yeah, I'm not doing special things. I just searched the cell which is attached to the glass surface.
How does it attach?
Mm. Yes, yes, I don't know exactly what kind of interaction is working between glass and cell membrane, but yes, I'm sorry, I can't answer that question.
Okay.